Okay, so this is chapter 17, and we're going to be discussing the special senses. So, special senses include the following. They include the sense of smell, vision, sense of taste, hearing, and balance. That's compared to general senses. The general senses, we can have the sensation of touch, vibration, pain, temperature. Okay, so what makes these general senses is that their receptors are found all over your body. So they are, we say general because they're not in one specific area. The special senses, their receptors are in very specific places. So, for example, the receptors for smell, they're found in your nasal cavity. Vision, those receptors are found in your eye, in your retina. Taste, those are found in your tongue and throat. Hearing is found in your ear, those receptors are in your ear, and balance is also found, those receptors are found in your ear. We will start by looking at olfaction. Olfaction is the sense of smell. The receptors are in the nasal cavity. And so the chemicals of objects that you're smelling move into your nasal cavity, and those chemicals are dissolved by the mucus in the nasal cavity. Once those chemicals are dissolved, that will stimulate the olfactory nerve endings. The olfactory nerve endings will converge into what we call the olfactory bulb. And then the olfactory bulb will bring those action potentials through the olfactory tract to the temporal lobe. And in the temporal lobe, that's where the olfactory cortex is. So if you remember when I was talking about sensations, all sensations, it doesn't matter if they're general sensations or if they're special sensations, they're all going to first go to the thalamus. And then from the thalamus, they synapse on a neuron that will go into their special cortex area. But I did say there was one exception, and that is olfaction. Olfaction, the sense of smell, does not synapse in the thalamus. It is the only sensation that doesn't. All of the rest of them have to synapse in the thalamus, and then they synapse onto other neurons that will go to their special areas. So here we see the sense of smell is going straight to the olfactory cortex in the temporal lobe without stopping in the thalamus. So here's the diagram. I just want to show you this. Here we see a rose, and those chemicals from the rose come into the nasal cavity. And then here in the nasal cavity, the nasal cavity is lined by a mucous membrane. and that mucous membrane secretes mucus. And so then the mucus breaks up those chemicals, and then those chemicals will stimulate the nerve endings. So right here are all of those nerve endings, and then they create action potentials in these little olfactory tracts right here. Those olfactory tracts lead into this bulb, which is called the olfactory bulb. And from there, those action potentials travel through the olfactory tract and directly go to the olfactory cortex in the temporal lobe. Next, we have gustation. Gustation is the sense of taste. So we know where taste, where our receptors are. Our taste receptors are found on our tongue. And the taste receptors are located in the taste buds. So the taste buds are found on the projections of your tongue and throat, and those projections are called papillae. So taste buds are found on the papillae. There are 4 different types of papillae. There are the filiform papillae, the fungiform papillae, the valate papillae. In your notes it might say circumvallate papillae, and the foliate papillae. The foliate papillae contain up to 1,300 taste buds, and that's quite a bit since the entire tongue contains about 7,900 taste receptors. The valate papillae contain up to 100 taste buds. The fungiform papillae contain about 4 to 5 taste buds, and the filiform papillae contain no taste buds, and they're really there just to create a little bit of friction so that you can move things around across the top of your tongue. Here's a diagram showing you 3 of the 4 different types of papillae. We have the valate papillae, and so you can see on this little projection on a little bump on the top of the tongue, you can see these little things here. These are the taste buds, right? Then here we have the fungiform papillae that normally contain about 4 to 5 of those taste buds. Here you have the filiform papillae that have no taste buds at all on them. And then what we're not seeing here in this picture are the foliate papillae. And the foliate papillae are going to contain, like I said, about 1,300 taste buds. So then let's take a look at a taste bud. So in a taste bud, so here's one taste bud here. And this taste bud then, of course, is on top of one of those papillae, one of those bumps on your tongue. And in this taste bud, we have what we call a gustatory cell. The gustatory cell has little taste hairs that protrude through those pores. So when a chemical comes in contact with those taste hairs, that stimulates that gustatory cell. Then the gustatory cell will stimulate a sensory neuron, and that sensory neuron will carry that information back to the cortex, and the special area of the cortex is called the gustatory cortex. And the gustatory cortex is found in the frontal lobe of the cerebrum. So there's several of these gustatory cells, each of them with their own taste hairs protruding through this open area, which we call a pore. And in between each of these gustatory cells is a stem cell. Because these gustatory cells don't last very long, they're going to die off, and then these stem cells will produce new gustatory cells. The sensory neurons will converge to form one of 3 cranial nerves. And those cranial nerves will be the ones that send the action potentials back to the gustatory cortex. So we have cranial nerve 7, which is the facial nerve. Cranial nerve 9, which is the glossopharyngeal nerve. And cranial nerve 10, which is the vagus nerve. So it's not what type of taste will determine which cranial nerve that information will travel back by. It's more location. So we take a look at the tongue. Here's the tongue, and behind the tongue is the throat. We can divide the tongue into an anterior 2/3 and a posterior 1/3. And then we have the throat behind that. So the cranial nerve that's going to monitor the anterior 2/3 of the tongue is cranial nerve 7, which is the facial nerve. So anything you are tasting that touches your tongue in this anterior 2/3 of your tongue, all of that sensory information will go back to the thalamus by the facial nerve, cranial nerve 7. Then you have the posterior 1/3 of the tongue. So any taste, anything that you touch to your posterior 1/3 of your tongue and are tasting back there, all of that information will go back to your thalamus through cranial nerve 11, which is the glossopharyngeal. Okay. And then we have the throat. So anything that you taste in the throat, and really the throat mostly has a lot of different, there's a lot of osmoreceptors, so it's detecting the presence of water, but there's also some taste receptors back there too. So anything that you taste back there, or the receptors for, whether if your throat's dry or not, that information is going to travel back to the thalamus through cranial nerve 10, which is the vagus nerve. Each of these nerves will bring their information back to the thalamus. Okay, and then from the thalamus, they will synapse in the thalamus onto another neuron, and that neuron will carry information to the cerebral cortex. And the special area in the cerebral cortex, again, is called the gustatory cortex. And that gustatory cortex is found in the frontal lobe. There are different types of taste. There is sweet and salty, sour and bitter. And there's also a taste for umami. Umami is a special taste. It's not really salty. It's not really bitter. It's Parmesan cheese is an example of umami, as well as like a broth, like a chicken or a beef broth. And then another taste is water. So these are the four main tastes, but then there's also umami and water. Next, we're going to look at vision. So we'll first start by looking at the external structures of the eye. So first of all, we have these things called palpebra. And so the palpebra are the eyelids. So we have the upper palpebra up here, and then we have the lower palpebra down here. So those are the eyelids. Then we have where the eyelids come together. They have to meet. So they come together and they meet on the medial side and then on the lateral side as well. So where they meet, that is called the canthus. So we have the medial canthus. This is closer to the nose over here. And then we have the lateral canthus, and that's closer towards the ear over here. There are also these little glands that are called tarsal glands. And so the tarsal glands are just on the inside of those, right on the inside of those eyelids. They're spelled tarsal, just like the bone in the foot. tarsal glands, and they help to secrete a fluid so that your eyelids don't stick together. Then if we look on the medial canthus, just on the inside of the medial canthus, we see this fleshy area that's called the lacrimal caruncle. A couple of other things that we can see looking at the external eye. You can see the whites of the eye, and the white of the eye all over here, it's called sclera. We can also see the colored portion of the eye, and the colored portion of the eye is called the iris. In the middle of the iris, you see this black circle here, and that black circle is called the pupil. And the pupil is actually a hole that goes into the eye. So the iris is surrounding that. So this is a colored portion is surrounding that. But you can also see that there's kind of a glare. If you look right here, there's a glare. And that's because there's a clear membrane over the top of the iris and the pupil. And that whole thing in there, that clear area that goes all the way across, is called the cornea. If we take all of the skin away, we can see some other structures. So again, here's the eyelids up here. This whole thing is the eyelids. You see the medial canthus and the lateral canthus and the lacrimal caruncle. Okay. Then we also see some things like this gland up here. This gland is called the lacrimal gland. So that gland produces tears. Right? So it produces tears. The tears then will flow through these little ducts and will empty onto the eye. And as a person blinks then, the fluid travels across the eye towards the medial canthus and then drains into this bulb container over here that we call the lacrimal sac. And then from there, the tears, the fluid, flows into this canal or this duct that we call the nasolacrimal duct. And that nasolacrimal duct will go all the way down to the sinus area. Right? So that's why when you tear up, you start to sniffle because all of those tears are draining into your nasal cavity. In this picture, we can also see these muscles that are called the extrinsic eye muscles. We'll learn in a minute what the intrinsic eye muscles are, but I want to talk about the extrinsic eye muscles. So the extrinsic eye muscles, we can see at least 4 of them here. They include this muscle right here, which is called the superior rectus. So rectus means straight. So this muscle goes straight back. That's why it's called the superior rectus, because it's superior on top of the eye and it goes straight back. Down here, this one is called the inferior rectus muscle. It's on the inferior aspect of the eye and it goes straight back. Then we have these oblique muscles, and oblique muscles mean that they don't go all the way back, straight back. So here we have the superior oblique muscle, and we can see it attaches. It's going to attach onto the sclera, and then it's going to hook around here, and then it's going to attach to somewhere in the bony orbit. So it's not going straight back, but it's still on the superior, the top of the eye. Down here then we see the inferior oblique, and that inferior oblique muscle is attached to the sclera, and then it goes back and attaches to the bony orbit. So all of these muscles are going to originate on the bony orbit, and then they're all going to attach to the sclera of the eyeball. So in this picture, we're looking at a cross-section drawing of the eye. And so I want to show you a couple of things. There's different layers on this eye. We're going to look at the layers. But first, let's go over some of those things that we already talked about to give us some orientation. So here is the white of the eye that we call the sclera. Okay, and then that goes all the way to the back of the eye. So we can see that sclera, the white of the eye, goes all the way to the back of the eye. And the white of the eye then, it attaches to this other cornea, which is like a clear membrane. And so there's that cornea right there. So that's that clear membrane I showed you that was kind of giving a sheen. And underneath that, it's clear so you can see the iris or the colored portion of the eye underneath that. So if we look underneath that, we can see this area right here, and then this area right here, and those are those muscles that we call the iris. Those are the pigmented, part of your eye, that circular part that's colored, and then the inside of that eye is that pupil, which is black, right? So here's this whole thing all in here, the colored portion of your eye, again, that's called the iris. And then over the top of the iris and the pupil is that cornea. Now, covering the sclera, at least a portion of the sclera, is a conjunctiva. And so the ocular conjunctiva, this is just a clear membrane. You see it right here. That is surrounding a little bit of the sclera. You can see the sclera underneath it. And that conjunctiva, that membrane, comes back a little bit into the bony orbit, and then it turns around like that, and it creates a little bit of a pocket. And we can see that on the bottom too. So here we have the conjunctiva on the bottom, and then it sort of works its way around. It's like an epithelium that covers first the sclera, and then it wraps around and it covers the inside of the eyelids. So where this membrane is covering the sclera, we call it ocular conjunctiva, and where it's covering the inside of the eyelid, we call it palpebral conjunctiva. So ocular and palpebral are just indicating the location of the conjunctiva. And the conjunctiva is a clear membrane. So you might have heard of conjunctivitis or pink eye. That's because this conjunctiva gets infected and gets really irritated, and it causes the blood vessels underneath it to get really red and swollen and irritated, giving the eye a pink appearance. So continuing to look at this picture, in between the iris, or right in the middle of the iris, we see the pupil, and that's an opening, that's a hole going back to the rest of the eye. And right behind that pupil then will be this crystallized structure called the lens. Right, so now we have these structures have kind of separated the eyeball out into different cavities. So the cavity that's in front of the lens, that is called the anterior cavity. And the cavity that's behind the lens, that is called the posterior cavity. So they're filled with different things. The posterior cavity is filled with a jelly, gelatinous type substance that we call vitreous humor. And because it kind of holds together like one big blob, we will call it the vitreous body. Then in front of the lens, the cavity that's called the anterior cavity, that's filled with more of a watery type substance that we call aqueous humor. So we have aqueous humor in front of the lens, and we have vitreous humor behind the lens. Now, the cavity in front of the lens, that anterior cavity, it can actually be divided up into two different chambers. There's the anterior chamber, and there's the posterior chamber. And they are separated by the iris. That's what separates it. So you can see, here's the cornea. So in between the cornea and the iris, this whole area in here, that's called the anterior chamber. And then a much smaller chamber between the iris and the lens here. It's much smaller. That is called the posterior chamber. And we have this fluid that's called aqueous humor that flows from the anterior chamber through that hole, that opening, the pupil, and then into the posterior chamber. Here's another picture that's showing the two different cavities. So the cavity in front of the lens, here's the lens. The cavity that's in front of the lens is called the anterior cavity, and the cavity behind the lens is called the posterior cavity. The anterior cavity then is divided up into an anterior chamber in front of the iris and a posterior chamber behind the iris. The other thing that this picture shows us in very good detail is that there are actually 3 layers to the eye. The layers can be called tunic or they can just be called layers. The outermost layer is called the fibrous tunic or the fibrous layer, and that's made up of the sclera and the cornea. So here's the sclera, this grayish area, and then that is continuous with the cornea, and that's continuous with the sclera on the other side, and that just wraps itself around the whole eye, right? So that's the outermost portion, the fibrous tunic or the fibrous layer that contains both the cornea and the sclera. On that sclera, the sclera again is the white of the eye. On that sclera, that is where the extrinsic eye muscles attach. It's kind of a tough outer layer that's just very protective. And it's white. The cornea is that clear membrane over the top of the iris. The cornea is going to help to focus light. So when light hits that cornea, that light will bend to go back to the lens. The second layer is the vascular tunic or the vascular layer. The vascular layer contains the iris and the what we call the ciliary body, and it also contains what we call the choroid. 3 things in that vascular tunic. We call it vascular because it provides a route for blood vessels into the retina. The retina is on the inside of the eye, and so that's why we call it vascular. The iris is actually a smooth muscle, and it's going to control the size of the pupil. We'll take a look at the 2 different layers of smooth muscles of the iris on another slide. The ciliary body controls the shape of the lens. So we're going to talk about that too on another slide. In the choroid are a lot of blood vessels, and the blood vessels are going to deliver things like oxygen and nutrients to the layer inside the eye, which is called the retina. The deepest layer of the eye is called the neural layer or the neural tunic, and another name for that is the retina. So here we see it's in the darker red area, right? Or it's an orangey area. And then in between that we have the choroid. So you can see all 3 layers in there. The retina contains what are called photoreceptors. So these are actually the receptors that detect light and detect color and detect detailed visual information. There's 2 different types of photoreceptors. There's rods and there's cones, and we're going to take a look at those in a different slide as well. And we're going to look at how those photoreceptors then activate sensory neurons and send that information back to the thalamus, which will then go to the cortex. We're going to look deeper into the eye at the retina, but before we do, I want you to look at the iris. Again, the iris is the colored pigment of the eye. So here we're seeing the iris. There's no pigment in there, so it just actually looks like a muscle, and that's because those iris layers of the iris are muscles. They're smooth muscles, which means that they are controlled involuntarily. So when we look at the muscles, we see that there's really 2 different layers. So we look at the outside here, and okay, so we're looking at this outside layer, and then there's an inside layer. So in the outside layer, we see that the fibers of the muscles are radiating out. And so we say that those muscles have a kind of a radial orientation in that muscle. And so when that muscle contracts, what it's going to do is it's going to pull the inside of the muscle here, pull it towards the outside of the muscle. In all directions that's going to happen. And so when that happens, the pupil then is going to get bigger because it's going to pull all of this towards the outside. So this is what we're seeing when the dilators are contracted, when those muscles that are in the radial formation are contracted. They have pulled out away from the middle, and now we see that the pupil gets a lot bigger. And that is called pupil dilation. Right? So in chapter 16, when we were talking about the sympathetic nervous system will innervate the dilator muscles, and it'll cause contraction. Then we look at the inner layer, the inner muscles in here, and we can see their orientation goes around and around. So when muscles have this type of fiber orientation, where the fibers just go around and around in a circle, we say that that's a sphincter muscle. And anytime we have a sphincter muscle, when the muscle is contracting, it is the inner part of it is going to move inward. And it's going to make the lumen or the opening much smaller. So on this side over here, we're seeing what happens when this muscle contracts. It bulges out like this. Right? That's what sphincters do. They get all big and puffy like that, and they bulge out, and they narrow that opening right there. So they narrow it, and when that happens, we call that constriction. So that's pupillary constriction. And so here we're seeing that the pupil constrictors are contracting. So a question that I would have for you on the exam would be something like, what happens to the pupil when the muscles that are in an radial orientation contract? And then you would say pupil dilation. What happens when the inner muscles that are in a sphincter formation contract? And then you would say, well, they constrict then, the pupil constricts. So these pupil constrictor muscles, the inner muscles here, they're innervated by the parasympathetic nervous system. So when the parasympathetic nervous system is stimulated, it will cause this constrictor muscle to contract, and the pupils will constrict. And so with the sympathetic nervous system, they're doing that because you're trying to rest, and they really don't need to take in the whole scene. They're not looking for predators. The eyes aren't looking for anything dangerous, so they can afford to constrict those muscles and contract the constrictor muscles and not allow as much light in. So here's my drawing of the same thing that I'm just showing you. So when the dilator muscles here, when they contract, we get pupil dilation. Right? So when the radial muscles contract, we get pupil dilation. And this is the sphincter muscles over here. So when the sphincter muscles contract, then you get pupil constriction. So now I want to explain how another muscle of the eye, of that vascular layer, controls the shape of the lens. So in this picture here, we can see the lens right here. So this is the lens right here. And it's connected over on the sides by these little strands that are called the suspensory ligaments. So each of those strands are the suspensory ligaments. Those are a structure that you have to identify on your lab list. And then the suspensory ligaments attach themselves to this muscle right here. And this muscle is on the outside of the iris, right? So we can see that on the outside of the iris. And that muscle is called the ciliary body. So here's the name of it, ciliary body. So when the muscle contracts and relaxes, it can change the shape of the lens. Now, a couple of things you need to know about the shape of the lens if this lens has a lot of crystals in it, but they're real fluidy type crystals, when you're looking at an object that's far away, that lens will become real flat. And that's so that it doesn't bend the object too far. So the cornea and the lens both bend light. As light comes to the cornea, then the light will bend a little bit, and then when it gets to the lens, it'll bend a little bit more, and that's so that whatever you're looking at is going to hit the back of the retina, and the retina is something we're going to talk about next, and there's a specific spot on the back of the retina that's called the fovea. So the purpose of the cornea and the lens is that they're supposed to bend light, so light will go back to that fovea. So by making the lens really thin, then there's not going to be as much bending, and that's when you're looking at things far away. They really don't have to bend a whole lot to get back to that point. But when you're looking at something up close, now that lens is going to have to bend a lot more, so the lens becomes real round. And so when the lens, in order for the lens to become round, those ligaments have to go really relaxed, all right? And then when the lens is going to become thin, then those ligaments have to become really tight. Now we have to think about this because the muscle here, the ciliary body, is not going to do what you think it's going to do. In order to make these ligaments that go all the way around, in order to make them really loose so that the lens becomes round, this muscle would have to contract. So when this muscle contracts, because it's a sphincter muscle, it bulges out and it pushes closer towards the lens, and it makes the suspensory ligaments then become real slack. So that's going to happen when we want the lens to become real round, because you're looking at something close up, right? So the ciliary body will contract, and the suspensory ligaments will become slack or loose. On the other hand, if the lens is needed to be really thin because you're looking at something far away, then those suspensory ligaments have to be really, pulled tight. And in order for them to be pulled tight, then that suspensory or that ciliary body is going to relax. Because if the ciliary body relaxes, then that muscle, that sphincter muscle, is going to be wider, and it's going to pull all of those suspensory ligaments more tight. So let me take a look. Let me show you a different picture. All right, so here we have the lens. This is the lens. Here and here. So if you remember, when the lens is flat, that means you're looking far away. So that means you're looking at a distance, right? That's distant seeing, distant vision, right? So when you're looking far away, it needs to be real flat, and that means that these suspensory ligaments have to be pulled really tight. And in order for them to be pulled really tight, then that sphincter muscle has to relax, because when the sphincter muscle relaxes, it's much thinner, right? It's just thinner, and it can pull then on those ligaments and make them really tight. But when you're looking at something close up and going from distant vision to close-up vision, that is called accommodation. So during accommodation, that muscle, which is called, again, the muscle is called ciliary body. So that's the ciliary body right there. When you are looking at something close up, that ciliary body contracts. And remember, when any sphincter type muscle contracts, it gets really fat, right? So it's bulging out. And when it gets fat and bulges out, then those suspensory ligaments get really loose, right? They get real loose like that. And then the lens doesn't get pulled tight. Now the lens is going to be bigger and fuller and more round instead of flat in distant vision. Here's yet another picture. So I'm just showing you, again, in the vascular layer, you have the ciliary body, which is right here. And then you have the iris, which is right here. And here's those suspensory ligaments. So on that lab exam, maybe you'll see this picture. And here are those suspensory ligaments right here, attaching the ciliary body to the lens. Okay. And then here's the iris, and then inside the iris is that opening that's called the pupil. This picture is great for a couple of reasons. You can see the anterior chamber. So remember, the anterior chamber is part of the anterior cavity. And then you can also see the posterior chamber right here. So you can see those really well, you know. Now, inside the anterior and the posterior chamber is aqueous fluid. It's a real watery type of fluid. And if too much fluid is being produced and it's not draining, it can bulge against the cornea of the eye. And then we call that glaucoma, which is a dangerous condition where people can lose their eyesight. So that aqueous fluid actually has to drain. Now the aqueous fluid is going to be able to go right through that pupil into the posterior chamber and then back again into the anterior chamber, and it can flow through both places, but then it has to drain. And so it's going to drain into this opening right here that we see in the anterior chamber. And it used to be called the Canal of Schlemm, but now we call it the venous scleral sinus. So that's what you're going to be seeing on your diagrams, the venous scleral sinus. All right, and this is the picture you would see in the lab exam. That's where the opening is. So now what I want to do is go and look at the innermost layer, which is called the neural layer. And the neural layer, another name for that is the retina. And this neural layer contains the receptors for vision. And we call those receptors photoreceptors. So when you look at the inside of the eyeball, that whole thing in there behind the lens, that is called the retina. And it contains the photoreceptors, right? So the photoreceptors, there's two different types of photoreceptors. We're going to look at both of those. There are the rods and there are the cones. And they both do different things, but they both allow you to detect visual information. So we'll look at those in just a minute. What I want to do is I want to show you, they're scattered, all these photoreceptors are scattered all over the back of the retina, and there is this one little indentation here that is called the fovea, and that's where most of the photoreceptors are found. So the macula contains a lot of cones. Cones help you to see real detailed objects. Now there's also neurons, sensory neurons attached to each one of those photoreceptors that can be stimulated by the photoreceptor, and all of those neurons are going to follow the outskirts of that layer, and they all merge together and exit from the eyeball at the same place. And where they exit, that place is called the optic disc. So where they're exiting, there's no retinal material there, so there are no photoreceptors in that area. So the optic disc, another name for that, is the blind spot. Because when any type of light is shining on this spot right here, you're not going to see it. You're going to have a blind spot wherever that is. And everybody has a blind spot. So I'll explain that also in a couple of minutes, the blind spot. But then those neurons, they merge together and they exit through that area that we call the blind spot. There's some arteries and veins that exit with it. But as it exits away from the eyeball, it then forms a nerve, that cranial nerve that we call the optic nerve. So the optic nerve is a big, thick band of neurons that will exit from the back of the eyeball. So here's a close-up picture, and we can see how all of these neurons are merging together and they are forming that optic nerve. And we can see that in this spot then, there are no photoreceptors. These cells here, out here, these are the photoreceptors. So you can see that in this spot, there are no photoreceptors, right? So that is that optic disc that's also known as your blind spot. All right, so now what I want to do is I want to show you, you can see there's three different cells in here. So we're looking at this, which is the photoreceptor, and then we see a bipolar cell right there, and then we see this neuron right there. So we want to take a closer look and see all of those. It's kind of weird because the light comes into your eye, it comes to the back of the retina, and then the light has to work its way through these cells to actually get to that receptor, the photoreceptor, and activate that. So let's take a look at another picture, a close-up. So here's a close-up of the retina, and so we can see two different types of photoreceptors. This is the innermost area. This is the inside of the retinal area. So when it's the back of your eyeball, so when light's coming through, it first has to get through this layer. It's going to work its way through these cells and through these cells, and then it's going to come over here and it's going to hit one of those photoreceptors and then activate it. And when those photoreceptors get activated, then they will stimulate this neuron right here. And this neuron is a bipolar neuron. Remember, it's bipolar because this is the cell body, and you can see that there's two poles coming off it, one on each end. Here's the dendrite, here's the axon. Then that bipolar cell is going to stimulate the next cell that we call the ganglion cell. And it's the ganglion cell, there's the cell body, it's the axons of the ganglion cells that are all going to form that, they're going to merge together to form that optic nerve. So it's kind of weird that the light has to filter through all of these cells before hitting that photoreceptor and then working its way back the other way again. I think, I've tried to do some research on that. I think that it's just a way of filtering out a lot of the light. But anyway, if we look at the layer where all those photoreceptors are, we can see that there are two different types of cells. We have the cones, which are less frequent, and we have the rods in between the cones, which are more frequent. Okay? So rods don't discriminate color. Rods don't discriminate color. So when your rods are active, you're not going to be seeing different colors. You're not going to be able to make out different colors. They also can't detect very detailed information. Okay, so they can't see either one of those things. They can't see colors and they can't see detailed information. So it's more like what the rods do is they allow us to see shapes and shades of gray in dim light. So those rods are really working in dim light. So if you're in your room and you turn off the light and you look around and you can still see objects, you're not going to see a real clear line around those objects, and all of those objects are going to look gray. None of them are going to have a color. But if you turn the light on and now light is able to, there's more light hitting those photoreceptors, those cones become activated in bright light. And so then when the cones become activated, they provide color. So they provide color, they give us a sharper, clearer image. And there's, like I said, there's a lot of these cones that are found at the macula, at the center of the macula, which is in the fovea. They also give us, besides color, real detailed images. So the rods are really working in dim light, and the cones are really working more in bright light. So here's what you can see at the back of the eye when you're looking through a scope. And so the back of the eye, this really white yellowish area right there, that's the optic disc. So that's where all of those ganglion neurons, the axons from them, are converging. So there's no photoreceptors there. Okay. And then just off to the side, just lateral to that, you see another area. That's the fovea. And at the center of the fovea is an area that's called the macula lutea. The macula lutea is the area where it has a high density of cones. So when you're looking at something directly, most of that light will be bent by the cornea and by the lens and directed back to that fovea, back to the macula lutea. So here's a picture. It's showing your two eyes, and so each eye has its own visual field, and then those visual fields overlap each other so that your brain can get information from both eyes. And so the light that comes in hits the back of the eye at the retina, the photoreceptors become activated, and that will eventually stimulate those ganglion neurons. The ganglion neurons then converge together to become the optic nerve. So that happens in both eyes. So the ganglion, the axons of those ganglion neurons, they are traveling down and they meet. The optic nerve from the right eye and the left eye, they meet at an area that's called the optic chiasm. And at the optic chiasm, some of the neurons from the right eye will go over into the left optic tract, and some will stay in the right optic tract. And if we look at the other eye, some of the neurons will cross over into the right optic tract, and some of them will stay on the same side in the left optic tract. So we get a crossing over of neurons so that your brain, both your right side of your brain and your left side of your brain, will know what is being seen in both eyes. Then those optic tracts lead back to the thalamus, which is what we've been saying all along. All senses go back to the thalamus, and then they synapse on other neurons. And then those other neurons that they synapse on are going to go back to that special area in the occipital lobe that we call the visual cortex. A couple of last things that I want to mention about vision. And first of all, you've heard of the term cataract. So a cataract is when that cornea, behind there's the iris, okay, and then there's the lens, right? So this is the pupil. So this is the cornea. And so when that loses its transparency, it's called a cataract. Sometimes it can be, you can really see that cataract on a person. It becomes whitish on the cornea. I also want to talk about normal vision. So you've probably had your eyes checked. Normal vision is 20/20. So that means that a person who's standing with normal vision is standing 20 feet away from an object, and they're being tested and compared to another person that's the same distance apart, 20 feet away. They can both see at the same clarity. They can both see just as good as the other. A person who has better than normal vision might, for example, have 20/15 vision. When they have 20/15 vision, that means a person who can see normally would stand 15 feet away from an object, and the person being tested can see just as clearly standing 20 feet away from the object. Then when we look at someone who has vision that's worse than normal, say 20/100, for example, that means that the person with normal vision can stand 100 feet away and see an object, but the person being tested can see that object at only, they have to go all the way up to 20 feet away in order to see that same object. A person that has 20/200, so a normal vision person can stand 200 feet away and see the object normally, but the person with 20/200 has to stand 20 feet away to see the object or to see the letters. If a person has 2200 vision, even when wearing contacts or glasses, then they are considered legally blind. And the last thing I want to say about vision is about color vision. So there are actually 3 different types of cones. So these cones, different types, they're going to detect different types of colors. So there's a red cone, and there's a blue cone, and there's a green cone. And when all 3 of these cones are stimulated, a person sees white. So when you're looking at the back of this screen, actually when you're looking at the light, like the sun or a light bulb, you're seeing white because there's so many different wavelengths of light that they're stimulating all 3 cones, and then you see white. And when you're looking at like a white piece of paper or this white screen back here, the light that's coming from the lamp or from the sun, whichever is giving you light today, it bounces off of this screen and then heads into your eyes, where it stimulates all 3 of those cones. On the other hand, a person that's looking at something that's black, when you're looking at something that's black, what's happening here is the light from the sun or the lamp, it is being absorbed right here by that dark color. And so nothing is bouncing back regarding this small area back to your retina. And so this small area then looks black because no cones are stimulated. So when you see white, all 3 cones are stimulated. And when you see black, no cones are stimulated. All right, now we're going to look at hearing. So we're going to look at the ear. So the ear can be divided up into 3 different sections. We have the outer ear, or otherwise known as the external ear. So that's going to include all these structures in front of these structures here. Anything in front of that is the inner ear. Then we have the middle ear, and so that's going to go from the external ear to the internal ear. And then we have the inner ear. So we want to look at all the different structures that are in each of these areas, and we'll start by looking at the external ear. So the external ear has, first of all, we see the ear that you normally think of, right? The ear there, that is actually called the auricle. That's the auricle of the ear, and it acts kind of like as a funnel, and it funnels in sound. So sound waves are funneled into this canal. This canal then is called the external acoustic meatus. So you don't want to call it the auditory meatus. You want to call it the external acoustic meatus or the external acoustic canal. Either one is fine. Now this is lined by a mucous membrane, and on that mucous membrane are these glands that are called ceruminous glands. Ceruminous glands secrete a cerumen, which is your earwax. So those are the glands that are going to build up that earwax. At the very end of the external ear, we see this membrane right here, and that membrane is the eardrum. Another name for the eardrum is the tympanic membrane. Okay, so there's the tympanic membrane, and so we have these sound waves hitting that tympanic membrane. On the other side of the tympanic membrane, we have the middle ear. Now the middle ear has these bones, these 3 little bones in here that we didn't talk about when we went over bones. But those 3 little bones in there are called the auditory ossicles. Auditory ossicles. So these, the incus, the malleus, and the stapes, and the one we want to pay special attention to is the stapes. So the stapes is S-T-A-P-E-S. It looks like a little stirrup. It looks like something, like a stirrup from a saddle. Okay, it's actually covering up an opening into the inner ear, and that opening is called the oval window. So underneath that stapes is the oval window. Now there's another structure here in the middle ear, and sometimes people say it's part of the inner ear, but it's really part of the middle ear. There's this long tube here going down into your nasal cavity. And that is called it's going into the nasopharynx. That's called your auditory tube. So this whole middle ear is lined by a mucous membrane. If mucus or fluid gets in there from swimming in a lake, or if bacteria get in there and mucus is being produced, all of that is supposed to drain down through the auditory tube and go into your nasopharynx, which is your throat. And then you can swallow it or spit it out. So it's supposed to drain there. Now, if it doesn't drain, then it will build up inside the middle ear and bulge against that tympanic membrane, and now you've got a middle ear infection, and that can be very painful. Children tend to get more middle ear infections than adults do because this auditory tube is more horizontal in infants than it is. As they grow, it becomes more vertical. Okay, so now let's look at the inner ear. So we said the stapes is covering up a window or an opening into the inner ear, and we call that the oval window. And just below that is another opening here. This one's not covered by a bone. This one's just covered by a thin membrane, and that is called the round window. So we have both the oval window and we have the round window. And we're going to see where they both lead into specifically, but what they are openings into this bony structure here that we call the labyrinth. And it's a bone, and on the inside of that bone is a membranous labyrinth that looks just like the bony labyrinth. And so it looks like this big snail creature here. So we've got this big head part of the snail, and we call that the vestibule. And then we have these antennas coming off of this snail, right? So there's 3 of those, and we call those the semicircular canals. Then we have the body of the snail, which curves around and around like this, and that curved part like that is called the cochlea. So there's 3 different parts to this bony labyrinth. And then the membranous labyrinth on the inside is identical to it. And so it kind of creates 2 different cavities. It creates a cavity in between the bone and the membrane. And then there's another cavity on the inside of the membrane. So we're going to look at that as well. Now here you can see the nerve. So we've got sensory neurons that are attaching themselves to receptors in these structures, and they are going to carry the information back through their axons through the vestibulocochlear nerve. There ends up being 2 branches here. So we've got a vestibular branch, and so let me explain what the vestibular branch is. This big area of the head, which is called the vestibule, the vestibular branch is coming from the vestibule area. Those nerve endings are kind of like at the ends of the semicircular canals, and they're also at receptors that are in the vestibule. And they come back, the axons extend backward, and they form the vestibulo branch of the vestibulocochlear nerve. Then we have the cochlea, and the cochlea has sensory neurons extending away from those receptors, and they join there. Those neurons are called the cochlear branch, and they join with the vestibular branch, and together they form that vestibulocochlear nerve. Going back and looking at a bigger picture of the middle ear, I want to show you 2 muscles that are found there. The first one's called the Tensor Tympani muscle. So this big muscle right here, which is called the Tensor Tympani muscle, it's attached to this bone right here, and that bone is called the malleus, and the malleus is stabilizing the tympanic membrane. So that muscle we call the Tensor Tympani because it's keeping tension on the tympanic membrane. Then there's a smaller muscle down here, and this one is called the stapedius muscle, and that one is stabilizing the stapes. All right, so let's look inside the bony labyrinth. So here you can see inside the bony labyrinth, and you can see this blue structure here. That is the membranous labyrinth. So we've got the bone on the outside, and then we've got the membranous labyrinth on the inside. Okay, and so there's a space then between the bone and the membranous labyrinth, and then the inside of the membranous labyrinth is hollow, so there's another space in there, and there's fluid then that travels through both of those areas, in between the bone and the membranous labyrinth, and then on the inside of the membranous labyrinth. So if we look on the inside of the membranous labyrinth and we go down to the base of those semicircular canals, we can see these structures in there that are called receptors. So there are receptors that are at the very end of those semicircular canals. The semicircular canals are arranged in a very distinct arrangement. So there's the anterior semicircular canals, or semicircular ducts, however you want to call them. And so then this receptor right here is going to be monitoring whenever fluid is moving inside the anterior semicircular duct. Then we have the lateral duct. So we see the lateral duct right here. And at the base of the lateral duct is a receptor. And the way it's oriented, right at the base there, it's going to detect any movement of the fluid inside the lateral duct. And then we have the posterior duct right here. And the posterior duct is arranged in a way that here's its receptors at the base of its semicircular duct, and it will detect any movement in the fluid inside that duct. So when does the fluid move? When does the fluid move in those ducts? Well, the fluid will move when you move your head. So whenever you move your head in certain directions, you're going to get fluid in one or more of those ducts to move. So for instance, with the anterior duct, when you nod your head, when you are nodding your head yes, then the fluid in that anterior duct is going to move, and it's going to stimulate this receptor, and the receptor then sends the information back to your brain, telling your brain that you're nodding your head. When you look at the lateral duct, the fluid in the lateral duct is going to move when you shake your head no. So when you are shaking your head from side to side and saying no, the fluid in that duct is moving and will stimulate that receptor, sending that message back to your brain, telling your brain that you just shook your head no. Then we have the posterior duct, and in the posterior duct, the fluid will move in there when you put your ear to your shoulder. So in other words, when you are laterally flexing your head and putting your ear to your shoulder, you're moving the fluid in that duct that will stimulate that receptor, and that information will go back to your brain, telling your brain that you're bending your head to your shoulder. On the next slide, I'm going to be showing you those specific receptors up close so you can see what they look like. Now, in addition to the semicircular canals, there's these other receptors here that are found in the vestibule. And those areas in the vestibule are called the utricle and the saccule. So we're going to talk about them collectively as both of them together. We're not going to separate them out. But those receptors in the utricle and the saccule, they're going to detect acceleration or deceleration. So if you're moving forward or moving backwards, the fluid is moving this way and that way, and they're going to stimulate those receptors and tell your brain that you're moving forward or you're moving backwards. They also will detect if you're going up or down. So in other words, they're affected by gravity. So if you're going up or down, that fluid in there is moving up or down, and that's also going to stimulate those receptors in the utricle and the saccule. So this is what the receptors look like at the end of those semicircular ducts. Here you have, these are the receptor cells right here, and they have these little hairs sticking off of them. Okay, so they've got these little hair cells, and these little hair cells are actually sticking up. They're kind of embedded into this jelly-type substance, and that's so that they're not going to become stimulated if any little tiny movement comes along. But if a bigger movement comes along because you're moving your head, then the jelly substance will move, and then that will bend those hair cells. And by bending those hair cells, it stimulates, it generates an action potential in these sensory neurons. Those sensory neurons, then the axons will all converge together, and they are going to form that vestibular branch of the vestibulocochlear nerve. So then we'll take a look at the other receptors that are found in the vestibular area, in the vestibule, the utricle and the saccule. And it's pretty much the same way. Here you have the receptor cell, and you have these little hair cells sticking off of the receptor cells, and then they're embedded in this gelatinous material. So they're not going to bend over the little bit of movement. And then sitting on top of the gelatinous material are these little crystals that are called otoliths. So all of these little purple things, they are little otoliths or little crystals. Now, they just add some weight down to this jelly substance so that, again, those receptors just aren't going to be stimulated over a little tiny movement. When there is enough movement, though, and these crystals are moved in a direction, they're going to move and bend those hair cells. Then those hair cells stimulate the sensory neurons, and the sensory neurons are all going to converge to form that vestibular branch of the vestibulocochlear nerve. So let's see this in action then. So when gravity is kind of pushing down on those crystals and keeping those hair cells from really, moving a whole lot. But when you tilt your head posteriorly then, the gravity is going to move that crystal in a direction, and that will move the gelatinous material in the same direction, and then that will bend those hair cells. Then those hair cells will activate that receptor, and the receptor will then generate action potentials in the sensory neuron, and those neurons will converge together to form the vestibular branch of the vestibulocochlear nerve, and then we'll head back towards the brain. Okay, now we want to look at the cochlea. So the cochlea is that body of the snail that I talked about that turns around. It's a series of tubes that just curl around each other. This is a picture showing the stapes. So here we have the stapes right here, right? This whole thing is the stapes, and it's covering up the oval window. So the oval window leads into this cochlea, and the membranous labyrinth separates the cochlea into two different ducts. So this first duct, a duct underneath that oval window, is called the vestibular duct. And the fluid is going to flow through that duct, that vestibular duct, and it keeps flowing through all the way to the end. And when it gets to the end then, it reverses direction at the tip, and it starts to go back the other way. And now it is in the tympanic duct. And so that tympanic duct winds its way around on the other side of the tympanic membrane, and it will exit then. It'll bounce against that window that we call the round window. So the round window will be over on this side. So I'm going to show you another picture where you can actually see this a little bit better. Okay, so here we have a hand-drawn cochlea, and I want to label some of the things here. So first of all, we have this membranous labyrinth on the inside of a bony labyrinth. So this right here, this is that membranous labyrinth. What that means is that it's a membrane, and on the inside of that membrane, it's hollow, and so there's a fluid running inside that membrane. We call this part of the membranous labyrinth the cochlear duct. And the cochlear duct contains a fluid called endolymph. So this separates the bony labyrinth into two compartments then. So we have sort of a passageway on the top and we have a passageway on the bottom. And so the passageway on the top is called the vestibular duct. And the vestibular duct is filled with a fluid called perilymph. So that fluid flows all the way up through that vestibular duct, and then it gets to the tip of the cochlea, and then it starts coming back. Now, as it comes back, we call that passageway a different name. So the passageway on that side, we can see it all the way, right? There's part of it, down here is part of it. And so we call this the tympanic duct. And the tympanic duct still contains that perilymph because nothing was really separating where the end of the vestibular duct met the beginning of the tympanic duct. And one other term I want you to be aware of here is that at the bottom of this cochlear duct, there is a basement membrane, and we're going to give a name to that basement membrane, and we're going to call it the basilar membrane. So it sounds a little bit like basement membrane. That's the basilar membrane. So the receptors for hearing are inside the cochlear duct. They are in the endolymph in the cochlear duct. I'm going to show you a picture of what that receptor looks like. It's called the spiral organ, and we're going to see what that looks like. But right now I'm just going to show you the location of it. So it will be periodically spaced in this cochlear duct. Right, so it's periodically spaced in there, and that's the spiral organ. In your notes, you might see it as the organ of Corti, but these days we're calling it the spiral organ. Let's look at the beginning of that vestibular duct. So at the beginning of the vestibular duct, there is this window leading into the vestibular duct, and it's covered up by this one of these auditory ossicles, and the name of that auditory ossicle is the stapes. So the stapes is covering up the oval window, right? So it covers that up, and then at the end of that tympanic duct, there's another window, and that window is right here, and that window is called the round window. And the round window is covered up by a membrane. So there's just like a little membrane covering that up like the top of a snare drum. So now let me show you what happens in the process of hearing. So this is your external auditory meatus right here. External auditory meatus, right? That's your ear canal. So these air waves come in. They're sound waves. That's what they are. Sound waves carried in the air. And they hit that tympanic membrane, which is right here. This is the tympanic membrane. And the tympanic membrane starts to vibrate, right? So it's vibrating. That causes the incus and the malleus and the stapes to start vibrating. So they're vibrating. Then when the stapes vibrates, it vibrates the oval window. So now the oval window is vibrating. And the vibration there on that oval window causes the fluid, that perilymph, inside that vestibular duct to start vibrating. But eventually that vibration of that fluid will hit the cochlear duct. And the vibration vibrates the whole cochlear duct. So this whole cochlear duct is vibrating back and forth, and it starts to displace the fluid on the opposite side in the tympanic duct. So now we've got fluid vibrating in the tympanic duct, kind of like pressure waves in the fluid. And then that fluid works its way over to the round window, and the round window can bulge out. So the round window bulges out and it kind of absorbs that pressure from the fluid, and then the fluid can stop. So we don't want fluid just constantly sloshing around inside these ducts. So that's how the fluid how the pressure waves begin and then how they end. But as those pressure waves are pushing through this cochlear duct, it is stimulating the receptors inside this spiral organ. And there are nerves, there are sensory neurons attached to this spiral organ. And those sensory neurons are going to converge with the cochlear branch of the vestibulocochlear nerve. The vestibulocochlear nerve is cranial nerve 8. So where those vibrations hit the cochlear duct will determine the pitch, like how high of a frequency will the sound have. So it will have a different frequency if the vibrations hit down here than if the vibrations hit the spiral organ way up here. So that's how that will determine the pitch. And then the loudness will be determined by how fast are those vibrations coming. So the faster the vibrations come, the louder the sound actually is. So let's take a look at that spiral organ. And I have the spiral organs down here below, so we can see them down here. Here is the spiral organ right here at rest when no sound is really coming in. So there is, first of all, we can see we have this down here. This is that basilar membrane. Okay. And then there's like this gelatinous, this jelly type of substance right up in here. And the receptor cells are kind of caught up in that gelatinous type material. So we see those, they're right here. So that is a receptor cell. There's a receptor cell, and there's a receptor cell. And they have these little hairs sticking off the top. So all the receptors for hearing and sight are little hairs, right? So those hairs are sticking up, and on top of that spiral organ here is another membrane right here, and that membrane is called the tectorial membrane. So we can see that the hair cells, though, when there's no sound coming in, they're straight up and they're kind of touching that tectorial membrane, but they're not bent at all. And they are attached to a sensory neuron, and the sensory neuron then, if it gets stimulated, all of the axons of those sensory neurons merge together again to create that cochlear branch of the vestibular cochlear nerve. Right, so they're all merging together. So what happens then in the sound, when we have sound waves coming through, the pressure waves come through and they hit this cochlear duct, and the cochlear duct, the pressure waves move all the way through the cochlear duct, and this whole basilar membrane then is going to bounce. So it initially moves down, and then the pressure comes back and it bounces back up. So when it bounces back up, it's going to take those cells that are right on top of it and push them upward. So they're pushing them upward, and then the hair cells that are coming out hit that tectorial membrane and they bend. So then those hair cells are bending. And what we've been seeing is that whenever those hair cells bend on any of those receptors, they're going to become activated. And so then when they become activated, they activate these sensory neurons. And now the sensory neurons have these action potentials, and they're going to send those action potentials back to the brain through the cochlear branch of the vestibulocochlear nerve. So this here is the spiral organ. And so like I was showing you in that other drawing, we have the tectorial membrane and we have the basilar membrane, and it's tucked inside that cochlear duct. So when fluid in that tympanic duct hits the tectorial membrane, the pressure will travel all the way through and hit that basilar membrane, and then it will continue on to the round window. But the basilar membrane then dips because it's going to bounce, and then it bounces back. And when it bounces back, here's those receptor cells with the receptor hairs. They will bounce up against the tectorial membrane, and the hairs will bend. That will activate and generate action potentials in these sensory neurons. And then these sensory neurons are going to converge to form the cochlear branch of the vestibulocochlear nerve. So this whole thing in here, this is called the spiral organ. So there are several spiral organs in that cochlea. So the cochlea is where the sense of hearing is detected. All right, so let's go ahead and go through the whole process of hearing, and then we're going to be done here. So first of all, let's look at the sound waves. The sound waves are coming in through that external acoustic meatus, and they hit the tympanic membrane, which is right here. So that's the first step. Sound waves arrive at the tympanic membrane. After that, then, the second step will be that the tympanic membrane vibrates and causes the malleus and the incus and the stapes to vibrate. And when the stapes vibrates, it's going to vibrate that oval window, because the oval window is right underneath it. So number 2, we have the movement of the tympanic membrane displacing the auditory ossicles, causing those auditory ossicles to vibrate. Then step number 3 is the stapes is vibrating, and it causes the oval window to vibrate, and that causes these pressure waves in the perilymph of the vestibular duct. And here you see it's called scala vestibuli, but a simpler term is just the vestibular duct. So that would be step number 3. Then step number 4, the pressure waves in the vestibular duct are going to distort the cochlear duct. And by doing that, they distort this basement membrane, which is on the bottom, right? So they distort that. And then the pressure waves head towards the round window, which is right here, and the pressure gets released. The fluid stays inside the duct, but the pressure itself gets released by bulging the membrane at the end of that round window. So that would be step number 4. Step number 5 talks about what happens inside that cochlear duct. When that basilar membrane vibrates, that will push up against those cells that are in there, those receptor cells, and then the receptor cells hit the tectorial membrane on the top, and the hair cells bend. And then finally, number 6, when those hair cells bend, they are going to stimulate the sensory neurons of the cochlear branch of the vestibulocochlear nerve. And so here we see number 6. Here's the sensory neurons that are coming from those cochlear ducts, coming from the spiral organs. And then this information is going to head back first to the thalamus and then to the auditory cortex. Okay, and that ends Chapter 17 on the special senses.